Hi class and welcome to Lab 8, which is on photosynthesis. This lab will take you through a worksheet, explain a experiment that we would be doing in class, and then have you answer some questions. For the most part, you can kind of figure it out on your own as you read through the worksheet, but I'm going to kind of take you through the experiments and explain them a little more in detail. You can use your textbook to make sure you understand these objectives and terminology. I would encourage you guys to watch the Chapter 7 lecture video and study that PowerPoint before doing this lab, and that might help you a little bit. The overall equation for photosynthesis is shown here, which you should be really familiar with by the end of this lab. Photosynthesis is what plants and certain bacteria are able to do with light energy. So they trap light energy and use it to synthesize organic matter or sugar. So plants will take in carbon dioxide and water, and using energy from the sun, they create sugar, which is the C6H12O6, oxygen, and water. Photosynthesis takes place in chloroplasts, where plant pigments absorb solar energy. And the principal pigment in advanced plants is chlorophyll, which absorbs light energy, specifically in red and blue portions. Hence the green color of the leaves of advanced plants. So they absorb, what they do is the chlorophyll absorbs light energy in red and blue and emits green, and that's why plants are green. In lab today, so we're going to be talking about chromatography, which is a technique that separates molecules from each other on the basis of their solubility in particular solvents. And this will allow us to qualitatively determine the presence of an accessory pigment in green leaves. So in lab today, you'll perform paper chromatography to note the occurrence of different pigments in green plants. Okay, so first, write a hypothesis relating the level of photosynthetic activity to light intensity. Remember that most hypotheses are written in an if-then format, meaning if this happens, then this happened. So your hypothesis should say something like, if light intensity increases, then there will be less or more photosynthetic activity. If light intensity decreases, there will be less or more photosynthetic activity. And you guys can choose your own. Below is a picture of our experimental setup containing a sprig of an Elodea placed in some water with 2% sodium bicarbonate solution. The sodium bicarbonate is the source for the carbon dioxide. And why is carbon dioxide necessary for this experiment? And I'll let you guys figure that out as you guys go through that equation of photosynthesis. We use an inverted funnel used to cover the Elodea, and an inverted test tube of water is placed over the funnel, making sure that there are no air bubbles in the inverted test tube. And this creates what we call a closed system. So if any gas is produced from the Elodea, it will be the bubbles will know it's coming from the plant and nothing else. And then we're also going to put a light next to our experimental setup to measure the intensity of the light to see how the intensity of the light has an effect on photosynthesis of this plant. So a lamp was placed within 20 centimeters of the photosynthetic apparatus, and the plant was allowed to sit for 5 minutes to ensure bubbles are being produced by the Elodea. And what gas will be produced in the bubbles? And for this, you should go back up to your photosynthesis equation, and you should see here what gas will always be produced by plants in photosynthesis. So the number of bubbles produced in 2-minute intervals over a period of 20 minutes was counted. And the number, so what we are doing here is we are counting the number of bubbles that are being produced every 2 minutes from this Elodea plant. And you have to write down the total number of bubbles after your 20 minutes. And then the average, that just means the average being produced at each 2-minute interval. And then here, so to find the average, you'll add them all up and divide by the total number. And then you'll do the same when we take our light source and we move the lamp or light source 50 centimeters away. And you can already see here, we're getting more bubbles when our light source is farther away. So you guys need to ask yourselves, is 20 centimeters or 50 centimeters further away from the experimental setup? When would there be more light intensity when the light is closer or when the light is further away? And then at which location, I'll let you guys answer these questions on your own and send me a message if you guys have any questions, but think through that a little bit. Now we'll be identifying plant pigments using paper chromatography. Plant pigments are colored substances produced by plants, and they're able to absorb visible radiation between violet and red. These plant pigments also give color to leaves, flowers, and fruits. And the main group of pigments found in plants are chlorophylls, as well as a variety of accessory pigments. These pigments can either absorb, reflect, or transmit various wavelengths, which we're going to see here. And chromatography is a technique in which molecules in a solution can be separated and visualized. Pigment molecules already have color, so they're very easy to see once they are separated. And the technique that we used in lab is to separate them, relying on their differences in molecular weight and solubility. So we placed a plant residue on a strip of paper and then placed it vertically in a large test tube with a small amount of solvent at the bottom. And as the solvent is absorbed into and up the paper, it will dissolve and carry the pigments with it. So the smaller and more soluble pigments will be carried up faster and further up the paper than the larger, less soluble pigments. And when the solvent has reached the top of the paper, the pigments will have separated out. So you need to identify the different pigment bands using the chart on the next page. This is the bottom of the paper that is pointed, and the top of the paper is at the top. And the smaller, more soluble pigments travel up the paper faster, and the larger pigments will stay at the bottom. So using these colors, you can identify the pigment name and the color on the strip. So the blue-yellow color would be this color. There's a bright chrome yellow would be way at the top. And then we have a olive or yellow-green color and a yellow or a yellow-brown color. So here's the yellow-brown color. And here, right, is the olive yellow-brown color. So then you need to enter the distance the pigment band travels. So if you can print this off or just use a ruler. And what you'll need to do is you'll have to measure it in centimeters, but put in as the number of millimeters. So 1 centimeter has 10 millimeters, 2 centimeters has 2 millimeters, and you need to measure how far the pigments traveled up the strip. From that, you can answer which pigment is the most soluble, traveled the furthest distance, so which is closest to the top, which pigment is the least soluble, traveled the shortest distance, which pigment is known as the main photosynthetic pigment. You might need to look at your textbook for that. And what is the main role of pigments in plants? And then the benefits to plants having more than one type of pigment as well. Then there's one more activity, the carbon dioxide fixation. This is during the second stage of photosynthesis. The plant takes up carbon dioxide and reduces it to a carbohydrate like glucose. So the carbon dioxide in the solution surrounding the LODA should disappear as photosynthesis takes place. Phenol red is a pH indicator, and when it is in acidic conditions, it will be yellow, and when it is in basic conditions, it will be pink. When we add carbon dioxide to a solution, it forms carbonic acid. So the pH of the solution will become acidic with the addition of carbon dioxide. So if the solution becomes yellow, it will be becoming more acidic because carbon dioxide has been added. So what gas is being added to the beaker? What you're doing is you're starting out with a beaker of phenol red solution, and it's pink or basic, and then you breathe into the beaker with a straw. So what gas is being added to the beaker from your breath? What color did this turn the beaker and why? And I think because once you figure out what gas you're adding, you should know what color that should change the beaker because of that gas you're adding. And the answer will be in this paragraph right there. Then we have set up 2 test tubes with phenol red that you turned yellow by exhaling into them through a straw. In one tube, you add a piece of Elodea, the plant, and the other tube remains empty. You place both in front of a light source and let it sit for 30 minutes, and the results are pictured below. So one turns pink and one turns yellow. So you can see here in acidic conditions, yellow is produced when carbon dioxide is added, and in basic conditions, it is pink. So describe any color change and which tube experienced the color change and explained what caused the differences you observed. So what causes it to change pink or yellow? And again, all of the information can be seen right here in this paragraph. So that takes us through this lab. This will be due on Sunday as well. We won't have lab next week because there's a week of spring break, and I'll also make an announcement of that. I hope you guys are all doing well, and thanks for listening.